So I will start my class. Am I audible? Hello?
Yes, sir.
Okay. So today we will discuss about how to do blast. So let me present my screen. So I had already opened up this particular site. The name of the address is ncbi. I'm typing it over here. ncbi dot nlm dot nih dot gov. So this is essentially the website. You can, if you want, you can write it down. This is the particular website if you want to go use this. Okay, once you open this particular website, you go to the right hand corner. There you will find blast. First let me allow. Okay, on the right hand side, let me increase the size. Let's see. On the right hand side, you will find this region known as blast. This link, you have to open this. So the new site opens. This is the blast site. So what is blast? So blast so this is blast over here. Blast essentially, it finds regions of similarity between biological sequences. The program compares nucleotide or protein sequences to sequence database and calculates the statistical significance. So essentially what it does, it takes a nucleotide sequence, it takes a or a protein sequence, and then in a particular format, what it does, it runs into the database. It searches against the database, and from in the database you have lots of billions of sequences present in the database. From that database, it will, what it will do, it will compare with your query sequence, the sequence which you are putting in for search. That is your query sequence. With that query sequence, what it will do, it will go into the database and it will search for more sequences which have got similarity, and based on the similarity it will give out a statistical result, an e value it will give. You will see that out of 500 amino acids, that protein sequence has got 500 amino acids, and you run it in a blast. You will see that out of this 500 you will find there are 450 of them are same and 50 of them are not same, and on the basis of the similarity it will give us statistically similar sequence. So the objective is, say for example, you have got the human HSF1 sequence. You have got a sequence. You don't know if it's a human sequence or you don't know what protein it is. You just got a sequence. Now you take this sequence, and when you put it in the blast it in the database, when you use this blast, when you blast it in the database, then that will give you a list of several sequences, sequences from different organisms, telling what sequence that is. It's an unknown sequence. Say for example, it's a monkey. You got a gene from sequence from monkey. That sequence, you run it in the blast, and immediately you will see that it will compare that sequence from monkey to that sequence. Even it can be monkey, human, man, everything, to all sequences present in the database. Say then you find out, okay, this is the sequence of a heat stress transcription. You see in the search that it's showing human HSF1, chimpanzee HSF1. You're seeing, okay, this is an heat stress transcription factor 1. So this the question there. Then you get confirmed that rather confirmed, you get an idea about what this particular sequence can be. Okay, this is a heat stress transcription factor, you get to know. Then you see 100% match with a chimpanzee HSF1. Then you will say, okay, great, this is a chimpanzee HSF. Okay, now I understand. So like this, it can be used for this is the basic purpose of this blast. That is, it compares nucleotide or protein sequences to sequence databases and calculates the significance. So comparing you have a query, the query sequence which you enter, it has to be entered in the FASTA format, and which we had seen in the last class what a FASTA format is. You have to enter that sequence in the FASTA format, and from that it will run throughout the database, and then it will give you your results. So what are the different types of blast? So you have a predominant, the most important two blasts you have to the two most important blasts which you need to concern yourself is the nucleotide blast and the protein blast. Apart from that, blast X, that is translated nucleotide, is you give a nucleotide sequence, and then you enter a nucleotide sequence, and then this nucleotide is automatically translated, and then this translated sequence is then blasted with a protein database which is present, and then you get your results. Blast N is protein, and it will give you a translated nucleotide. So you give a protein, and then it will back calculate, and it will make it into a nucleotide sequence, and then for that nucleotide sequence it is again blasting the throughout the genome. But the most important blast are nucleotide blast, where nucleotide sequences, DNA, RNA sequences are then blasted with a database of nucleotide sequences, and you get your results. And protein is with this protein. Nowadays you can see that there is a new primer blast in polymerase chain reaction, PCR, which you have read. When you design these particular primers, then you see if that primer actually binds to the sequence or not. You've designed a primer, so you paste the primer sequence, and then on the basis of similarity the primer will then bind to your DNA, the DNA which you want to amplify, the forward primer and the reverse primer. It will bind, and then it will then you can see for PCR amplification, then you can see that these two primers essentially they are perfect. But then suddenly you see that you design a primer and then you see that nothing is working. You're not getting any match, or you see the 5 or 6 nucleotides are not matching, or 5 out of 30 nucleotides, 5 of them are not matching, or 6. It's not a good primer. So you redesign your primer properly. So before you spend the money in synthesizing the primer, you check through this blast here. You can see, use this primer blast, and you can see if it is fine or not. So the name of the blast means basic local alignment search tool. This is what is meant by blast. Now let us go, we remember, let us open a sequence. If we remember, we had in the last class we had a sequence. The sequence file which I had sent you. I have here. Let us copy the in FASTA format. Let us copy the human HSF1 sequence and say if you don't know, this is human HSF1. This is an unknown sequence. You say copy this and let us do up. This is these are the protein sequence. Let us do a protein blast. You click this protein blast and you can see that you have blast P, or let's increase the size. You see blast N, blast P. So you have blast P. You can enter an accession number. Also, all these we will see what an accession number is. That is an identifier number in the database. If you give a few digit accession number for HSF and all the genes, they have got an accession number. If you search by that give this accession number, you will be able to retrieve this sequence from the database. So first thing is you can do a search HSF1, human HSF1, and then you'll see 101,000 sequences. Out of that you find human HSF1, and from there you copy out your accession number and keep it safely with you. And then later, whenever you want to retrieve the sequence, use that accession number and search in this NCBI site itself. And then once you search in this there, you will eventually you will find only the sequence which you need. You can do it as many times as you want. Even inside this, we will see in this accession number later on that you can find all sorts of information about that particular protein. Whatever information you need about that protein, you can find lots of information over there. So we entered the query sequence. Our query sequence is the sequence with which we want to inquire, which we are going to blast in the whole database. So I paste the particular sequence. Now you have a place known as query subrange. Say this is a 529 amino acid we had seen. We want to just see from amino acid number 10 to amino acid number 50, or 1 to end, whatever you want. This is 529 base pair amino acids, 529 base pairs. So you can do this, but for the first search let us not do keep this blank, because let us do it for the whole sequence. You can give this name. You can have it aligned 2 or more sequence. So it will find different sequences in the database and align 2 of them. Let us not do bother ourselves with this at the moment. Now we go to the database, non-redundant protein sequence, the whole database. You can use different sequence select proteins, that is the proteins which have been sequenced. They are it's a sequence. Then you have reference proteins, particular model organisms if you want. So whatever database you want, let's go to non-redundant protein sequences. Let us look at that. You can use organism. Let us see, it can be any organism in the database. Let us do. You can even restrict it in there. And here it's blast protein, protein blast you have given. Better to say show results in new window, or let it be in this window itself. So let me just refresh it. It's for some reason or the other it is not working properly. Let's keep show results in the new window first, and let's paste the sequence for here. The same thing I'm doing. I'm giving a job title, and I am pressing blast. A new window is opening. It is searching. This is the request ID, job title. It's searching. Now America is awake, therefore it will take some time. When America is sleeping in the morning, it works much more faster. So this blast P is working right now. In 12 seconds, 13 seconds, you have to now wait patiently what happens. You have to rub your hands like this. You can scratch your nose. You can adjust your glasses. You can wait for some time till your results come out. It will come out, you don't worry. It may take some time, but it will come when it's searching. So already 35 seconds have passed, and still now 53 seconds have passed. We are still searching. It's waiting. Now America, we are doing it at this particular time of the day. It will take some time.
Sir,
Yeah. Please
sir,
ask.
what does statistical significance mean in the definition of blast?
How significant is the similarity between these two sequences? Like in you are comparing, what do you do in a statistical test? Say for example in a t-test, you can compare a sample population, say how many people have got COVID, sample population with that of the India's population, of the whole population, a sample with the whole population. So you are seeing whether the number of people affected by COVID is the same as that of the whole population in India. Then you can say, okay, the trend is the same, the ratio is the same. So the number of people in India, the people having COVID in India is the same as that of the West Bengal. You can say that population mean and your sample mean, they are the same. So this is what you mean by statistically significance. Whether it gives the statistics, what does statistics do? It gives a confidence to the number or data which you obtain. It gives some level of confidence, right? That is the reason why you are doing this test. You have a test, you are seeing, okay, say for example, let us talk about a chi-square test, which you people have already done, the statistical tests which you have done. In a chi-square test, you have a field where you have say 1,000 plants. You have got perfect ideal expected ratio to get 3:1 ratio, Mendelian ratio, that is you need to get say for example 750 is to 250. That would be a ideal 3:1 ratio. But you don't get that 3 is to perfect. Say you get 700 plants and you get say 200 plants. 700 is to 200. Now can you say that whether this particular population it follows a Mendelian ratio, whether it follows a 3:1 ratio? Now you see, oh yes, maybe 100 plants have died over here and 50 plants have died over here. Whether is it significant? So you don't have any scientific way to say that okay, it is following Mendelian 3:1 law. So when you use a chi-square test, use this formula and see the distribution. in this particular non-parametric test, if you see a particular the distribute, so, after doing this chi-square test, you get a value, you get a probability p-value, a probability value. Now you see if this probability value is greater than 0.005 or less than 0.005, then you can say that okay, well, it is following Mendelian law. So you are getting a particular confidence that okay, it is following, it has got this much amount of error, but it is following a Mendelian hypothesis. So, and that is the null hypothesis which we are following. If it doesn't follow, say for example, you get 500 is to 500, that is 1:1 ratio. Oh no, it's not following a Mendelian law. So this is what you mean by statistical significance. So you give a sequence, 100 nucleic amino acid sequence is going, and that is also bringing out another 100 amino acids from different organisms in throughout the database. So and they are aligning it. So statistically, say for example, there are 100 amino acids, you see 10 or 15 amino acids are not matching. So how will you say that okay, it can be a different organism but they are the same gene? You don't have any particular scientific logic in saying that it is the same gene. So a statistical test automatically runs in this BLAST, and this statistical says okay, 10 are not matching, but it has got a 90% similarity. So once you see a 90% similarity, well, it is the same gene in a different organism. Say a similar gene, it belongs to the same family, you can say. Even if it's 100% match, then it's the same gene. If it's not 100% match, 99% match, it could be a polymorphism, that one amino acid change, or it could be something, it could be a different, very closely related gene. For example, in animals you have, in plants you have 20, in Arabidopsis especially, you have 21 heat stress transcription factors. And out of that, they are divided into 3 groups, group A, group B, group C. So group A, among the group A1 group, you find 95 to 97% similarity. HSF-A1D and HSF-A1B, if you see, you find the similarity. So these similarity tests are done to give us statistically significant scientific logic saying that they are similar. Am I clear? বুঝতে পারলে?
Fine. Yes, sir.
Okay. So now while we are explaining, this particular page opens up your BLAST. Now let us have a look, and what do you see over here? The BLAST results. This is the ID and this is the result. Search report you are getting. Human HSF1 529. This one was that. So BLAST P you have done. Query, molecule type you have given, amino acid 529 amino acids. Now what you can find over here? Description. So first you see heat shock protein 1. We have given the heat shock protein 1 of human. So this you find. Now this is its accession number. Say I open it in a new tab and let's see what it is. So you get all details on the heat shock protein factors. You can get the FASTA format, graphics. So definition, heat strength, 529 amino acids. In the database, everything most of the things which you need to know about it. You can get everything. Homo sapiens, reference, then who are the authors who found out. These are things, the name of the publication, which journal first given, then different remarks, different database, what are the different publications, titles of the publications, authors. You can get all the details over here. Then you can summary this. The product of this gene is a transcription factor that is rapidly induced after temperature and binds heat shock promoter elements, HSC. So all these things you can get datas. Then you can get the protein data, region. and you can get the, at the end of all this you will get the protein sequence. This is the protein sequence you get. This is the protein sequence basically which I had retrieved. I had the accession number, I retrieved this sequence and I have pasted this in the text file which you have seen. This was pasted in this particular file. This is actually taken from this. You can see AMD, LPV, you can see AMD, LPV. So basically I took it from here. I made it, converted it into FASTA. And nothing, I don't have to do much to convert it into FASTA. I simply have to go to the top of this page where it's written FASTA. And I get this sequence. This is the one which I pasted basically. Now, nowadays with a lot of COVID things are seeing, this is from gene peptide. Now why are they asking surveys now? I'm teaching. So you can have whatever you want, accession list, gene pept, FASTA, graphics, revision history, all summary. You can get lots of whatever things you want, you can get it from information from here. So this is, let's close this window and get back to the original place. This is the accession number which you had seen. And you see what are the things which you can see. You can see 100% match and a percentage, and you can see an e-value. E-value, the higher the e-value, the lower the similarity. 0 means very good, 100% match. So for the first 100 it's all 0. You can see beautiful match. You have got 91, 89, you have got different heat check from different monkeys. All these monkeys you can get. We will see. Even if we may find, oh, Bos indicus, cow is also here. Haha. So moose musculus, our mice, all their heat stress transcripts. So with this human thing, let's see moose musculus. It has got an 89.62% similarity. The mouse HSF1 and the human HSF1 has got an 89% sequence similarity. So that's pretty common. Then if you see horse, Icus, Pantroglotitis, that is your chimpanzee. With chimpanzee you have a 93.90. We will see, look at the alignment of how, what the similarity is gradually in this. So you can see that this is something which you get. This is the statistical score which you get here. So these are the statistical results which you get, and an accession number. Now let us look at a graphic summary of the I have asked only for 100. If I want, I can ask for 50 also. So it will reduce to 50. Let's look at 50 only. You can even select columns which you want to see. Maximum score, you don't want to see them. You don't want to see e-value. You can even select what you want to do. You can download for FASTA sequences, CSV file, whatever format you can download it. But I am not touching right now because I am teaching at the moment. Now let us look at graphic summary. So all the whole red means this part is not matching. This most part is more or less similar. Statistically they are similar. This is the graphic summary. Then let us go to the taxonomy. You have a different taxonomy. These are primates. All sequence of primates. Then you have rodents coming over here. Then you have ungulates. Ungulates like horse. Artiodactyla, Perissodactyla, all these ungulates. You have them coming over there. So this is a taxonomy you can get. Right. Then you can, let's see alignments. Let us talk about the alignments. So first, with your query sequence, query is the sequence which you did in a BLAST. You can see 529 out of 529, 100% similarity. Gaps means where there is no similarities. Where suddenly a gap is there. There is one gap in the multiple alignment. You see, there were lots of gaps where it's not continuous. So the first line is the query and the subject is the one which comes out from the database. From the database it comes out, human heat shock factor 1 comes out. It's basically blah blah. It's the same thing which is coming out. So gap means there is no gap, so it's basically the same thing. Let's go for the next one. From a different sequence of the same thing. Here also 529 by 529, no gaps, it's the same thing. Next one is also the same thing, 529 by 529, no gaps. Similarly, let us go to something where we will find gaps. Oh, we go to Hyalobates moloch. What is Hyalobates moloch? Let us have a look at what is Hyalobates molochs. It will be interesting to learn something of zoology also at the moment. It's the silvery gibbon. This is Hylobates moloch. We are talking about its HSF1. Now if we are looking at, you have it's basically 525 out of 529. 525 are similar, but there are no gaps, there are differences. For example, here there is this one amino acid difference over here. This plus sign is over here. You can see this, instead of this M you have a V over there in Hylobates, and here you see a gap. Instead of a V you have an A over here. It's not a gap actually, you have a different amino acid. Instead of valine you have an A over here, aspartate. So basically there is there are 4 differences which you see, no gaps. It's very common. Like that you go to Pan-paniscus. This is also a type of monkey, primate. This is also a type of gibbon, Nomascus leucogenes. You can see here, you see 526 out of 529, 99% similarities. Let us say then gorilla, with gorilla 526 by 529, lots of similarity with gorilla HSF. Let's see with chimpanzee. Chimpanzee 525 by 99% similarity. This is the similarity with chimpanzee. Here the difference, instead of methionine there is a valine. Everywhere in these monkey primates there is this difference. So it's a specific character, this methionine over there we are finding. So by this you can check all the alignments. Let us see if for one gap. The lower we go, do we find, here you see a gap, 28 nucleotide gap. With whom? With Papio anubis. Let's see who is Papio anubis. This is the olive baboon. So you with this olive baboon you find a gap. It's not so similar. They have got this extra 28 amino acids inside. The rest is aligning, but here you have a gap. This is known as a gap. So this is how you interpret your BLAST results. Now you can also get a phylogenetic tree if you want from these results. Let's go to the top. You can get distance of tree results. Let's see what's opening up over here. Just wait. Okay, so it is giving on the basis of all the sequences present, you can see an alignment. This phylogenetic tree is coming, showing which is close to which or what. You can get a blast tree view. You can get a multiple alignment if you want. We had seen a multiple alignment with Clustral. You can see it with Cobalt also. It's calculating multiple alignment. All these accession numbers, these are the accession numbers. Instead of writing, if you give the numbers, this is good as same. Let us say I will teach you. Let us copy this. We will copy this accession number sometimes. So you can see all the alignment of this protein. Let's take this accession number. How to search for accession number? Let's keep it copied. We will go to that. So this is essentially you can get a alignment also. Let's look at a phylogenetic tree. With multiple organism, it has been found out. So this is we have to let us not go to the details. We have to reset the tree, and we have to it's using Cobalt multi, but Clustral W is the best. So let us not worry too much about this. We can go back to the blast results. We have gone back to the blast results. So this is the blast results. Now let us, this is the end of blast. Now let us go how to search. I had copied this particular sequence. So let us go to NCBI. So we open NCBI, all database. You simply paste this over here and give search. It's searching all database, and you see you get the heat shock protein factor. I gave this accession number, and you see the gene pep, this the whole sequence you can get over here. This is how you use your accession number in order to get data. I can also go similarly, instead of that, say I don't have any information. Instead of accession, I write human HSF1, all database search. So it will give human HSF1. You can get this. Here you are getting the nucleotide sequence also. You can get all information, genome annotation, all details you will get. You can ask for human HSF1 protein. instead of full report you can go to gene, you can get the full report. So this is how you actually come with the search results. So with this I end my class. I hope you people could understand. Do you have any questions? কোয়েশ্চেন আছে, বুঝতে পেরেছো?
No, sir, no questions.
Okay, okay, fine. Then I am ending my class here.